So I'm going to be talking this morning about barcodes, molecular barcodes, and their applications for deciphering the biological circuitries and microbial pathogens, but also to identify the mode of action of drugs. So functional genomics is a field of molecular biology that allows you to explore genes and protein functions and interactions on a global scale. And what I mean by global scale is at the level of the whole genome. Okay? So we require the availability of fully sequenced genomes. In 1996, the first eukaryotic genome sequence has been published by André Gaufreau and co-workers here in a manuscript entitled Life with 6,000 Genes. So this reflects the size of the yeast genome, Saccharomyces cerevisiae. And this was followed by publication of the genome sequence of mouse and human by Craig Venter in 2001. So now, how could we use sequencing, genome sequencing data to in functional genomics? Well, by several approaches. Actually, we can understand the evolution of the species, okay, by doing comparative genomics and looking at morphology and synteny between different species and understand how these species evolve. Another approach is population genomics, allowing to understand mutation dynamics and disease transmission between population and within population. So this could be done by analyzing SNPs, single nucleotide polymorphisms, okay, through the use of whole genome sequencing or DNA sequencing. Another approach is transcriptomics, using RNA-seq technology, and this allows you to understand cellular responses to perturbations. You could combine transcriptomics with genome-wide location technology that allows you to understand the dynamics of gene regulation by looking at the location of transcription factors or chromatin remodeling proteins on the whole genome. This is called ChIP-seq, chromatin immunoprecipitation coupled to high-throughput sequencing. We could use microarrays as well in this approach and this approach. And the last approach is genome-wide phenotyping, or chemogenomics. So this allows you to understand gene function, but also to discover pathways and phenotypes, right, through the use of mutant collections. So I'm going to focus on this approach. Genome-wide phenotyping requires the availability of biological resources. So this is critical to have biological resources such as mutant collections, knockout, overexpression collections, as well as genetically engineered strains that allow to generate those collections, and tools for genetic perturbation. So you have many providers, for instance, Fungal Genetics Stock Center, Euroscarf, or ATCC, allow to get those resources and use them in your research. So high-throughput phenotyping is also an integral part of the emergent field of systems biology, right? It's called phenomics. So phenomics basically allows to identify the full set of phenotypes of an individual. So it's like a challenge, actually. You use this approach for assigning a relationship between a genotype and a phenotype. So it's a very complex question and challenging question. So now the simplest way of constructing biological resources is to set up gene deletion libraries and gene overexpression libraries, right? So gene deletion libraries allows you to perform loss of function screens, then get loss of function phenotypes. On the other hand, you could use gene overexpression to perform gain of function screens and get overexpression of phenotypes. And by combination of both approaches, you could get a phenotypic maps. And here you have 6,144 mutants of Saccharomyces cerevisiae. So this is baker's yeast. Every mutant is arrayed on that plate, and you can detect here that one mutant here is hypersensitive, doesn't grow on a medium. This one again, and this one, etc. You could have some resistance, so strains that are resistant to the condition used in the screen. So this is to say that yeast is a powerful and versatile tool, model organism, and a cool tool in molecular genetics. So it is easy to manipulate, right? And it's characterized by its short generation time, inexpensive use, and powerful genetics. So the genomes of these three yeast models has been entirely sequenced. So this is an advantage. And the genomics tools are available. And we also have rich resources to perform high throughput phenotyping in those organisms. Right? So they are used in many applications. For instance, Saccharomyces cerevisiae here is used in industrial applications in biotechnology together with Pichia pasteuris. Candida albicans is an excellent model for looking at processes related to infectious diseases and host pathogen interactions. Whereas Saccharomyces conmate fish and yeast, also called micromammal, is an excellent model for looking at processes occurring in higher eukaryotes, such as mammalian cells. So it's a nice model for looking at cancer research or research on mitotic processes as well, right? Cell division. So of course there is a variety of resources available. For instance, Saccharomyces cerevisiae, you have Euroscarp and ADCC provide you with around 6,000 strains, each carrying a deletion of the 6,000 cores. You also have overexpression collections provided by Dharmacore, and this collection was established by Brenda Andrews at the University of Toronto. And as POMI, you have rich resources as well. So you have heterozygous deletion mutants, as well as haploid mutants, that are available from Pioneer. And you also have overexpression collections available from Reichland Bioresource Research Center in Japan. Now for Candida albicans, we lack an overexpression collection. So this is certainly being generated by the groups of Christophe Denfer, Institut Pasteur in Paris, and Carol Monroe from the University of Aberdeen in the UK. But we still have a heterozygous diploid collection consisting of around 5,000 strains. So these are available from Leah Cohen Lab in Toronto, and they were constructed by Merck Frost Canada. So I'm going to talk to you later on about this collection in one of the applications. So I'm going to focus on this yeast, Candida albicans. So it's a diploid yeast. Its genome has been entirely sequenced, consisting of 6,200 cores approximately, and a cycle genome of 15.5 megabases. So it's a polymorphic yeast. So it might switch from a yeast form that allows colonization of the GI tract to a high-fall form. This is a filamentous form, pathogenic form that allows to invade tissues and cause disease. It's part of the microbial flora, mainly gastrointestinal tract, but also the vaginal flora. And it's an opportunistic yeast causing both superficial and invasive infections. Among the superficial infections, you have oral candidiasis, so you have patches of Candida albicans in the oral cavity, but also onychomycosis, so this is infection of the skin that surrounds the nail. Okay. Then you have more severe infections, for instance, deep-seated infection in the oxide, such as the ocular candidiasis here, and also the development of biofilms. These are structures of highly adherent cells that develop around medical devices and catheters, and they are highly resistant to antifungal agents. So the treatment of candidiasis relies on administration of antifungal drugs. You have three main classes here, systemic antifungal drugs: polyenes, echinocandins, and basals. Polyenes, an example, you have amphotericin B, that targets ergosterol, which is the homologue of cholesterol in mammalian cells. Ecocandins, you have caspofondrin. Caspofondrin targets FKS1, beta-D-glucan synthase, so you have perturbation of the cell wall. And azoles target the 14-alpha linosterol This protein, encoded by ergolavin, so is involved in the biosynthesis of ergosterol. Okay. So azoles are fungistatic, whereas polyenes, for instance, are fungicidal. But treatment fails in immunocompromised patients in particular due to drug resistance and drug toxicity, especially during long-term treatment. Right? So understanding Candida albicans biology and pathogenesis allows us to basically try to identify new drug targets and develop new antifungal agents. There's also pressing need for biological resources and mutant libraries to perform functional assay in this yeast. So we initiated in 2009 in the lab of Christophe Denfer in Paris and Carol Monroe in Aberdeen, a project that was funded by Wellcome Trust with the goal of generating 6,200 Candida albicans strains, each overexpressing a distinct dwarf. Right? So it's a big challenge actually, because due to the size of the genome, so you have 6,000 genes. And this relies on the gateway technology from InVitrogen, relying on recombination-mediated cloning of every dwarf in an entry vector, right? So through BP reaction, so you amplify your dwarf and you flank this dwarf by attachment V1, V2 sites, allowing recombination to attachment V1, V2 sites in entry vector. And then this allows you to sequence the dwarf to make sure that there are no unintended mutations that were introduced during the PCR process. Then you use an R reaction to transfer your dwarf from the entry vector to the Candida albicans overexpression vector, carrying a tetracycline promoter, inducible promoter. And you place your dwarf here, and then you add doxycycline or anhydrotetracycline or tetracycline to induce high levels of expression. Then you come here with STO1 and you transform your parental strain. It's integrated in the genome of Candida albicans. So, Oscar Wilde was saying, nothing succeeds like excess. So what are the benefits of gene overexpression? It mimics gain-of-function mutations. It induces pathway activation. It complements loss-of-function phenotypes, so knockout phenotypes. It allows to study actually both essential and non-essential genes, so this is very powerful. It also bypasses the effect of genetic redundancy. So in case you have a multi-gene family, okay, you knock out one member, another member might hide your phenotype that has similar function. When you overexpress every member, you can get directly your phenotype, right? So this is advantageous as compared to knockout strategy. So by the time I started my postdoc with Christophe in Paris, we initiated a pilot project. So by generating 600 strains approximately, so 10% of the genome. So we've I've cloned 600 dwarfs, including transcription factors, kinases, phosphatases, and genes involved in DNA recombination and DNA repair. And we inserted in the vector, in each vector, a unique 20 base pair sequence, okay, called molecular barcode. So this allows you to identify every strain, okay, from the population. So it's serving as a strain identifier, and it allows you to quantify strain abundance in mixed population experiments. So you basically pool all of the set of strains in the same culture, and you grow them competitively. Okay. Then you extract your genomic DNA, and every barcode is flanked by common regions, sequences, allowing to amplify the barcode from the genomic DNA. So for every gene that was cloned, you have the corresponding barcode sequence. Strain 1, strain 2, another barcode sequence, and strain 600. Different sequence again. So how does the system work? So you pool your strains and you grow them competitively in the presence and absence of doxycycline for a number of generations, certain number of generations, might be 20, 40 generations. Then you extract genomic DNA, you amplify your barcode with the primers shown here. Then you label your barcode fragments with Cy5 for the treated sample and Cy3 dye for the untreated control. These are fluorophores. And then you mix your fragments and you hybridize the microarray, carrying the complementary sequence to the barcodes of the collection. So if an overexpression of a certain genes is toxic to the cell, for instance, this red strain here, you would have depletion of the strain from the pool during growth. So you would have less of the Cy5 signal. And this appears as a green spot on the macro, showing that you have higher abundance of the red strain here than in here. So then you scan your microarray, you filter your spots, you subtract the background, then you normalize your data. So you get the Cy3 signal on the x-axis, Cy5 signal in the y-axis. And you can see already that we have a bunch of strains that are sort of sensitive to treatment with doxycycline. And by looking at the usable gene, you find that many of those genes encode regulator of cell cycle progression and filamentation as well. Saying that when you overexpress genes important for cell cycle progression, there is a signal to say, okay, we should stop or decrease our growth rate. Okay. So these strains are basically overexpressed genes that confer decreased growth in the diabetes. And this was validated also in a screen by Saccharomyces cerevisiae done by Brenda Andrews. And we get also the orthologs in Candida albicans that cause slow growth when they are overexpressed. So this validates our strategy, basically. Now the good thing is that you can apply this technique or technology for the discovery of drug resistance genes. Okay? For instance, you could grow your pool with 40 microgram per ml doxycycline for 2 hours. So you induce overexpression. And then you treat or not your pool with gaspofungin. This is an antifungal drug that targets FAS1, so it perturbs the cell wall. And so you grow your cells for a given number of generations, you extract genomic DNA, you amplify your barcodes, you differentiate label with Cy5, Cy3, and you mix, then you hybridize to microarray. So if a given gene, the overexpression of a given gene confers resistance to gaspofungin, you would see that the strain carrying this gene has increased, will increase its abundance and increase the number of barcodes from that strain, and this appears as a red spot on the microarray. So we got, out of the 600 competing strains, we got some genes whose overexpression confers gaspofungin resistance out of that. Top 2 was previously shown to be a determinant of gaspofungin susceptibility. When you knock out POP2, you sensitize the cell to gaspofungin. Now when you restore one copy of POP2, you restore growth on gaspofungin. Another example is PGA59 was previously shown that a knockout, so this is part of multi-gene family, 62 and 59. When you knock them out, you have resistance. You activate resistance to gaspofungin. Now when you overexpress PGA59, you increase susceptibility. So it's the other way around. And we see here we validated two genes whose overexpression confers resistance to caspofungin, PGA37 and PGA19. So you see that they're able to grow on caspofungin containing medium as compared to control medium. So it's a nice strategy for looking at genes whose overexpression confers resistance to antifungal agents. To look at the drug mode of action. This is a integral part of the pharmaceutical drug discovery pipeline. So the drug mode of action is initiated at the beginning of clinical phase of drug development, at the phase of discovery, but also later on during early clinical development to look at the mode of action of a given drug in humans to see if you could have some side effects, for instance, or toxic effects. Right? So many approaches allows you to discover the drug's mode of action, including transcriptomics, for instance. You could use transcript profiling in response to drug treatment. You treat the cells with your drug, and then you look at the transcriptome changes in the mRNA of your genome, and then you could look at pathway potentially involved in response to the drug. Then you could use biochemical assays, for instance, affinity chromatography followed by mass spectrometry. So you derivatize your drug and then you incubate it. So you stick it to a matrix, you incubate it with total extracts, and then you wash and you elute the complex of your drug with the protein that binds to this drug. And then you could use mass spectrometry to identify the drug target. Computational methods, in silico docking. This, maybe you would see this with Hossein later on in his talk. High throughput imaging, this is a challenging technology. It's called high content screening. You could use microscopy basically, and you label the compartments, all of the compartments of the cells, the vacuole walls, reticulum, Golgi with different fluorophores. Then you treat with the drug and you look at the mode of action. So this is technically is very demanding. Okay, so you need expertise to do this. So it takes several images, and you have to combine all the images and see if the drug would alterate vacuoles or alter the nucleus, etc. Then chemogenomics. So phenotypic profiling with mutant collections. I'm going to focus on this aspect here. So in candidalbicans, this has been developed by Merck Frost Canada. They generated 2,800 candidalbicans heterozygous deletion strains with double barcodes. Number one, number two. This serves as an additional control. Okay, if one of the barcodes shows up, the other one should also show up. And then Canada is deployed. So they replaced one copy of the two alleles with the triple cassette, and they kept the second allele intact in the cell. So in these strains, you have already 50% of the protein pool that is decreased. Then if you treat with your drug, and the drug would inhibit the protein encoded by the gene X, you would sensitize the strain to drug treatment. And this would be the target of your drug, right? So the same principle, basically they competitively grew the heterozygous deletion strains in the presence of an antifungal drug, here as a proof of principle, fluconazole. Okay, but they did it for several biomolecules and natural compounds. And so fluconazole, as I was saying, targets ERG11, which is involved in the biosynthesis of ergosterol. Okay, so this leads to steroid depletion and accumulation of toxic ergosterol derivatives. So this will stop cell growth, but will not kill the cell. So this is a fungistatic nature of azoles. So after growth for 20 generations, you could extract genomic DNA, then you amplify the barcodes and differentially label with side ice. So, for instance, if a given strain is sensitive to fluconazole, you could get less of the Cy5 signal and disappear as a green spot on your microarray. So at Merck, they had this result that shows right away that the most sensitive strain is a strain that carries heterozygous deletion for the target of fluconazole, right, ERG11. But also they uncovered other genes whose heterozygous deletion causes sensitivity, including, for instance, CDR1, which is a pump. This is an ATP binding cassette transporter that expulses the azoles outside of the cell. So you could see that heterozygous deletion of CDR1 confers susceptibility also to fluconazole. So it's a nice technology for looking at the mode of action of antimicrobial agents. Now, you could combine both approaches, the gene deletion libraries and gene overexpression libraries, to look at the mode of action of drugs. So if you have, for instance, a candidate as an antimicrobial agent, you could now look at the mode of action through this approach here, by looking at mutant collections and overexpression, knockout collection and overexpression collection in combination. Now, the last approach I would like to talk to you about is to apply this, the barcode technology in the discovery of pathways involved in microbial pathogenesis. For instance, you know, K. is able to colonize the gut of humans following treatment with large spectrum antibiotics. Right? So we looked at, if we could identify genes whose overexpression affects K. fitness in actually a mouse model of GI tract colonization. So Volc female mice are fed with drinking water containing gentamicin and streptomycin. This allows you to deplete the microbiota, bacterial microbiota, and help in establishing the colonization by Candida albicans. Then you treat with doxycycline or not, control. This allows you to induce gene overexpression. During 4 days, then you gavage your mice with an inoculum carrying 5 × 10⁷ cells from your pool. Okay. And then you establish colonization for 10 days. Then you collect your stools, you homogenize them, and then you extract total DNA. Then you amplify your barcodes with the primary I showed you earlier, and you label with Cy5 dyes. Then you take the inoculum and directly extract genomic DNA from it, and you amplify barcodes and you label with Cy3. And then you mix and hybridize to your microarray. So if a given gene confers, the overexpression of a given gene confers increased colonization of the gut by Candida albicans, this would appear as a red spot on the microarray, but will not appear in the minus lux control. Right? So this is an additional control to make sure that what you have is really specific to overexpression. So using this technology, we were lucky to find only one hit, crazy 2. Overexpression of crazy 2 confers increased colonization of the mouse gut. Present in dust-dox treated mice, absent or abundance unaltered in the minus lux control. This is based on 2 independent experiments with n = 9 mice for a dox plus dox condition, and n = 8 mice for a minus dox condition. So what is crazy 2? It's actually a transcription factor of the C2H2 family that is homologous to crazy 1 from Saccharomyces cerevisiae and Candida albicans. Crazy 1 is involved in calcium signaling and also in the regulation of calcineurin pathway. But crazy 2 is not involved in this process, and we don't know its function yet. And it appears to have arisen through duplication of a common ancestor to crazy 1 and crazy 2. And it's specific to the Candida lineage. So specific to species that colonize the gut of insects and colonize the gut of humans. So we find this very attractive, and we've looked at the function of crazy-2 in KDAL by combining two technologies. The first one is chip or chip-seq. Okay. The second one is RNA-seq. Since crazy-2 is a transcription factor, so we could locate crazy-2 on the genome of KDAL and look at the genes that it directly regulates. So the principle is that you tag the transcription factor with an epitope tag, right? And then you treat your sample with formaldehyde and you prepare total extract. So this induces covalent links between the protein and DNA. You take a picture right away, instantly, and of the binding event of your transcription factor to its target. Then you prepare total extracts, you shear DNA by sonication to get approximately, on average, 200 base pairs. Then you immunoprecipitate with an antibody that recognizes the epitope tag. So you pull down your transcription factor with the DNA to which it binds. Then you reverse the cross-links and you purify your DNA fragments. So you need to know which DNA fragments are bound to your transcription factor. And then you sequence those fragments to look at the binding location of your transcription factor. So you see here that there are peaks, enrichment peak, upstream of your direct target. So you could combine this with expression analysis, RNA expression analysis. So you activate your transcription factor in the same conditions that those used in the chip experiment. You reverse transcribe cDNA, and then you sequence or you use microarrays, for instance. So you could combine both technologies, the list of genes that are bound to those that are upregulated or downregulated by perturbation or activation of your transcription factor. So the list of genes that is located here would be the genes that are both bound and upregulated by your transcription factor. Whereas those genes here are the genes that are both bound and downregulated by your transcription factor. So these would be the direct targets that are modulated, transcriptionally modulated by your regulator. Then you could build your transcriptional secretary based on the results that you have here and look for additional analysis. For instance, you could identify the motif to which your transcription factor binds, but also do gene ontology or gene set enrichment analysis to look at pathways that are overrepresented among the genes that are regulated by your transcription factor. Then this allows you to predict the function of your regulator and then test it experimentally. So we've tagged crazy-2 transcription factor at the N-terminus, and we placed it downstream of a tetracycline-inducible promoter. And we performed a chip-on-chip and transcriptomics analysis with two time points, 2 hour and 4 hour, following tetracycline treatment. Then we combined the data together. So what this is telling us is that crazy-2 binds and upregulates the expression of 103 genes in the genome, whereas it downregulates directly the expression of 63 genes, suggesting that crazy-2 acts as both activator and repressor of gene expression. So among the targets that we found that are upregulated, you see here adenosine transferases. These are involved in protein glycosylation. Okay. And then you have cell wall and cell surface proteins that are off, suggesting that crazy-2 might modulate the cell wall protein, the glycoproteins at the cell surface of KDAL, and this might trigger increased colonization of the mouse skeleton. So the hypothesis is that crazy-2 could have a role in efficient protein glycosylation and efficient production of cell wall proteins. We also did gene set enrichment analysis and combined the data with cytoscape to look at the processes as a whole, as a network. So the red spheres indicate enrichment of functional categories among the upregulated genes, whereas the blue spheres indicate enrichment of functional categories among the downregulated genes. So this is telling us already that among the upregulated genes you have enrichment of processes pertaining to interaction with the host. Okay, interaction with the host, adhesion to host, biological adhesion, pathogenesis. We also see right away cell wall components that are enriched among the upregulated genes. But interestingly, the hypoxic transcriptional program is overrepresented among the genes that are upregulated. And at the same time, you have downregulation of aerobic respiration. So this is telling us that when you overexpress Crazy2, this gives a signal to the cell as if it was growing in a hypoxic environment, in an oxygen-scarce environment. So based on the data showing that Crazy2 directly controls the expression of malignant transferases, so involved in N-glycosylation, cell wall proteins, and based on our finding that Crazy2 appears to adapt to hypoxic environment, we looked at tunicamycin susceptibility. Tunicamycin is a specific inhibitor of N-glycosylation rate. Under normoxia 30 degrees, but under hypoxia 37. So this condition mimics the gut environment where you have low oxygen levels, 37 degrees Celsius. And as you can see, the overexpression of Crazy2 with 3 independent mutants increases susceptibility to tunicamycin, whereas deletion decreases susceptibility to tunicamycin. Shown here that there is a high level of N-glycosylation occurring there, so it sensitizes the cells to tunicamycin, whereas here you have low levels of N-glycosylation. So our working model is that Crazy2 adapts to the hypoxic condition at 37, turning off cellular respiration, activating hypoxia genes, and modulating the cell wall glycoprotein expression levels on the cell surface in the molecules to confer increased GI tract colonization. So this aspect might give you an idea of how we could combine 3 subfields of functional genomics. First, with phenotypic profiling in the gut, we uncover Crazy2 transcription factor. Then we combine transcriptomics and genome-wide location to identify the targets of Crazy2 and make hypotheses on its function and how it confers increased colonization of the GI tract. So the take-home message here is that molecular barcoding allows you to perform phenotypic assays in mixed population experiments called competitive growth. So you could grow all of your set of strains in the same soup, okay, in the same flask, in the same animal. You don't need 600 different flasks. Or put them in animal to do animal experimentation or in vivo assays. So it's an economic approach. It uses highly sensitive readouts, so you have microarrays or bar-seq. You could sequence the barcode and quantify the abundance of every strain out there. And it allows to detect subtle phenotypes. So I hope I convinced you that barcode technology has many applications. It allows to identify molecular mechanism of drug resistance, as we've seen with classical function, to decipher the mode of action of drugs and growth in drug compounds, as with the Merck-Frost collection. And also allows to decipher the circuitry of microbial pathogenicity, as in the case of Crazy2 transcription. So I thank you for your attention, and I'm happy to